Hello, we're going to take a look at Chapter 18, Objective 1. Chapter 18 is over blood. So just real quick, let's take a look at the objectives over the entire chapter before we jump into Objective 1. Regarding blood, we're going to take a look at the primary functions of blood in Objective 1, its fluid and cellular components, and its physical characteristics. Objective 2, you need to identify the most important proteins and other solutes present in blood plasma. Number 3, describe the formation of the formed elements of blood. Formed elements, we're talking about the cells, red blood cells, white blood cells, and platelets, and how they are formed. Number 4 talks about red blood cell structure. Objective 5 is talking about white blood cells. Number 6 is talking about platelets. So we've kind of taken each one of those individual cellular components and broken them down separately. And then number 7, you need to know the ABORH blood groups. The ABO blood group, that should say an O right there, not just AB. There should be an O right there beside the B. And then number 8, discuss a variety of blood disorders. Okay, so let's just jump on in here to Objective 1. And we're going to be talking about the primary functions of blood, the fluid part and the cellular part, and the physical characteristics. Blood, if you remember, is a type of connective tissue. You've got 4 tissue types, and connective tissue is what blood will fall under. Blood is made up of 2 main components. If you remember with connective tissue, it was made up of cells with a lot of matrix, a lot of extracellular matrix between the cells. Now, that matrix could be a fluid matrix, which is what blood has. The cellular components, the cells that make up this connective tissue blood, they're also called formed elements. They're going to be red blood cells, white blood cells, and platelets. Red blood cells are also known as erythrocytes. White blood cells are also known as leukocytes, and platelets are also known as thrombocytes. So make sure you know the everyday word and the more correct term. And then what is the matrix made up of? That extracellular matrix. Again, this is a fluid matrix that exists between the cells called plasma. And what is the function of blood? Well, it kind of depends on what cell we're talking about, right? So primary, it does deliver oxygen and nutrients and removes waste from the body. This has a lot to do with the red blood cells. Other functions include defense. That's going to be kind of your white blood cells. Distribution of heat. That's going to be all of the blood doing that. And then maintenance of homeostasis. So in other words, your sensory mechanisms, your sensory receptors are picking up information about pH of blood, how much sugar is in the blood, how much sodium is in the blood, and then making how much calcium is in the blood and making changes accordingly. The composition of blood. So let's look at it a little more specifically here. So with the composition of blood, I'm just going to get my little laser pointer out here. Composition of blood is going to be 55% plasma. So you can see here in this test tube right here, you can see that the plasma is going to be this yellowy substance on the top. And then you're going to have this very, very thin, it's called a buffy coat right here. It's kind of represented in blue, and it's kind of a whitish color there in real life. Maybe it has a little bit of a blue tinge, but not much. This area is going to be the white blood cells and platelets, and that actually makes up less than 1% of blood are the white blood cells and platelets. So they're not very, the concentration is low for those 2 cellular components of blood. And then look at the amount of red blood cells in comparison. 45% of blood is made up of red blood cells. Now, within this plasma up here, you've got a lot of stuff that's going on in that plasma. The fluid portion of the blood contains proteins. About 4% of the plasma is proteins. You don't have to know specifically the percentage of proteins, but you should know what proteins we're talking about. Predominantly, it's going to be albumins. You also have some globulin proteins that are present, and you have some fibrinogen that's present. So those are the proteins that we're talking about there. Plasma is made up of 50% water. So at least half of plasma is actually water. And then you have these other solute particles. These are going to be your dissolved solids, such as, your sugar, your sodium, things like that. When we're looking at the white blood cells and platelets, Again, not very much, right? So this is less than 1% of blood is going to be white blood cells and platelets. If you remember, platelets are part of your clotting mechanism. They help clot the blood if there's a break in a capillary or a blood vessel. And leukocytes are representing all white blood cells. So it's a umbrella category, so to speak. So you've got leukocytes, and then you've got various types of these leukocytes or white blood cells. Specifically, neutrophils, lymphocytes, monocytes, eosinophils, and basophils are the specific white blood cells that are found in plasma. I should say in blood. Now, one way you can remember the order of how much you have of each of these in the blood is if you remember the phrase, never let monkeys eat bananas. That will help you remember, oh, I've got more neutrophils than I do basophils. So it just kind of helps. It's a little saying there to help you remember the order of those white blood cells in the blood. Okay, so let's take a look at why you would maybe have the red blood cells at the bottom and maybe have the plasma at the top. So obviously, if you look at blood, it's going to be red, okay? But if you let it sit, if you put it in a test tube and let it sit, what's going to happen is the red blood cells are going to sink to the bottom. The plasma is going to be on the top. And again, these white blood cells and platelets, that buffy coat's going to be in between the two. So why does blood settle this way? Why aren't the red blood cells on top, or why doesn't it stay mixed? This is a solution. It's a heterogeneous solution, so that means kind of like water and vinegar or oil and vinegar. You shake it up and then it separates back out. Unless blood is continually moving, it's going to separate out. Okay, it just is. So why is the red blood cells? Why are they going to be on the bottom? Well, they're just more dense, just like anything else. If you take rocks and water and shake it up, guess what? The rocks are going to go to the bottom and the water is going to be at the top. I could have said sand and water. Why? Because sand or rocks just weigh more than water does. It's less dense. So red blood cells, they're pretty dense. They have iron in them, that heavy metal iron. And so that's going to cause them to sink to the bottom. So blood testing, one way that you can measure how much red blood cells a person has is called a hematocrit. That's a test that is used to determine how many red blood cells a person has. Normal, it's going to vary for gender and age, is going to be about 36 to 50% should be red blood cells. Okay, and if you look over here at our picture, you can see that you have, maybe slightly less red blood cells than you do plasma. Again, that's going to average about 45%. So that's the number I'm going to ask on the test. Again, so 45% falls between 36 and 50. And again, that's going to vary. That 45 could be less or more based on gender and age. So this is actually blood that has been taken in a test tube. You can see right here, the blood has been allowed to settle. So you just put it on the counter and don't touch it for about an hour. And this is how it's naturally going to settle. So you're going to see this plasma, notice it's kind of yellowish in color, is going to be at the top because it's less dense. See this little area right here? That's that buffy coat that I was talking about. That looks like a buffy coat. It's got your white blood cells and platelets in there. And then here, the dark red, well, that's going to be your red blood cells. You can see that the blood in this next test tube is before it has settled, right? So that's probably right after it was taken from the patient. Now, based on the amount of red blood cells that settle, you can kind of get an idea just by looking. You can see that the red blood cells look like they're about equal in volume to the plasma. So this person's probably in good shape. Again, kind of like test tube number 1 right here. So you should have a little bit less red blood cells than the plasma. And again, it's kind of hard to gauge this exactly without any marks on the test tube, but you kind of get an idea. It's pretty close. Now, a person who is anemic, when that blood settles out, you can see the red blood cells, they're just not as, the amount isn't as much. So you can kind of get an idea that that person is anemic. And then you can see right here, well, there's a whole lot more red blood cells than there are plasma. This is called polycythemia. Poly means many, cyto means cell. Themia is referring to those red blood cells in the blood. So this would be an elevated hematocrit. And this isn't a healthy thing either. You wouldn't want That would be what you would maybe call thick blood. So that wouldn't be good either. That's hard on your heart. So, what are some characteristics of blood? The physical characteristics. One of them is it's more viscous than water. So, when we say the word viscous, what are we talking about? Well, we're talking about thickness. So, for example, honey is thick, syrup is thick, motor oil is thick. You know, when you pour it, it's very thick. Blood is that way as compared to water. So, about 5 times more thick or viscous than water. It's got a temperature of about 100.4 degrees Fahrenheit. Now, keep in mind, body temperature is 98.6. So, why would blood have a higher temperature? Well, that's because it's squeezing through these blood vessels. And if you were to take your hands and rub them together, what you're doing is you're putting two things together and that creates friction. Well, when these blood cells are passing through the capillary or an artery or a vein, they're bumping up against the inside walls. And when they do that, when they hit the inside walls, that generates friction. Friction builds up heat. So that's why your temperature inside the blood vessels is higher than normal body temperature. Now, the difference between oxygenated blood versus deoxygenated blood is bright to dusky red. So you can see right here, this is a bright red. This is going to be blood that's in an artery. This is full of oxygen. It's going to have a brighter red color. Now, you probably have not seen arterial blood. I hope you haven't seen arterial blood. This is not the blood they take when you go to the lab and get blood drawn. When you go and get blood drawn in the lab, they're taking venous blood, which is going to be a darker red color. And when you look at your blood vessels in your arms where they're probably taking the blood from, you can see they kind of look like they're a little bluish. Again, you can see right here, blood is not blue. So, but that dark red sometimes appears is so dark. Sometimes it appears like a purpley color through the as you're looking at it through the skin, through the walls of the blood vessels. It can appear this bluish, purplish kind of color, but it's very dark red. Now, arterial blood, they're not allowed to take that at the lab. That's going to have to be performed by a specialist doctor or someone along those lines. Because again, if you make a nick in an artery, it could cause you to bleed to death. So that's a little bit under more controlled circumstances, controlled environment. pH is normally 7.35 to 7.45. Keep in mind on your pH scale, this is going to result in slightly basic or alkaline. Remember, your acids on your pH scale go from 0 to 7. 7 is neutral, and then 7 to 14 is basic or alkaline. And your pH of blood is going to be slightly basic or alkaline. Just males are going to have a little bit more blood, 5 to 6 liters of blood compared to females, 4 to 5. Why is that? Well, males are just bigger. They're going to, again, they're larger individuals usually, so they're going to have more blood. To give you an idea what we're talking about here, because I know we're not familiar with the metric system as well. So if you've got a gallon, that's going to equal a little bit less than 4 liters. So you're looking at about a gallon to a gallon and a half, maybe about a gallon and a half of blood. So if you see those movies on TV, those horror movies, where they've got blood gushing everywhere, that's actually a lot more than what a person actually has in their body. So they're taking a little bit of liberty there. Now, in your packet that you purchased from the bookstore, you should see questions, in-class questions for Chapter 18. And this is question number 1 out of that question set 1 because it's over objective 1. I'll give you a second to take a look at that and answer the question. Okay, the 3 cellular components of blood are red blood cells, erythrocytes, thrombocytes, also known as platelets, and white blood cells, and that would be B, leukocytes. I'll give you a second for question number 2 in this set. So if you remember our picture, remember it was plasma on top, then the Buffy coat, white blood cells and platelets, and then the red blood cells. But if you remember, there was more plasma, 55% was plasma. Then you had red blood cells coming in at about 45%. And then the least amount that you had were the white blood cells and platelets. Remember, those made up about less than 1%. So your order should be 3, 1, 2. So that would be C. I'll give you a second for questions 3 and 4 in that set. Okay, number 3, the hematocrit test measures the percentage of what cellular component of blood? Well, that would be erythrocyte, red blood cells. And then the pH of blood is going to be slightly basic. Remember, it's going to be 7.35 to 7.45. So that should have been C. Let me go back up to here to number 3 a minute. So again, like I said, when we were going over the names of the cellular components of blood, you need to know them by both red blood cell, RBC, or erythrocyte, platelet or thrombocyte, white blood cell, WBC, or leukocyte. So you need to know their everyday terms as well as their anatomical terms. They're more biological names. And that concludes objective 1 for chapter 18.